Hi, I'm Peter Cook. Welcome to the pan-genomic model, part 2. In the first section, I showed that DNA loops stabilized by the transcription machinery are major structural motifs. I'm now going to consider a major force driving looping. And for this, we go back to what I will call the classical mechanism. If we consider a transcription factor present at a typical concentration and able to dimerize with a typical KD, we find that the equilibrium is very much to the left, with very little in dimeric form. However, if DNA is also present, which has two binding sites 10 kb apart, one and then the other might bind, and then this creates a high local concentration. One is tethered to the other through the intervening DNA, and so they're much more likely to interact and dimerize, and so stabilize a loop, which will remain for as long as one or other of the factors remains bound. Now, this classical mechanism, perhaps surprisingly, works through positive feedback to build an emergent bridging-induced attraction. So Davide carried out these simulations with those simple assumptions. He considers, for example, a long string of blue beads and two different transcription factors. Red ones can bind to pink beads and green ones to light green beads along the string. Neither binds to the blue beads. Sometimes the red and green factors switch between binding and unbinding states, which is known because many factors, for example, are phosphorylated and dephosphorylated to make them bind and unbind. And we need to include this to explain the rapid exchanges that are seen after photobleaching. Now, sometimes these particular spheres are going to represent a factor with a polymerase. And when, for example, this one binds, we will say that that particular bead will be transcribed. And this is what the final frame in a simulation might look like. There are so many blue beads that you can't see what's going on. So I'm now going to show you the same frame with all of the DNA fiber removed. And you can see that underneath it all, we've got clear clusters. And I'll show you frames. So we begin with unbound factors diffusely spread throughout the field. And then as we run the simulation, you can see that clusters are spontaneously forming, red ones and green ones, and that they're clearly rarely mixed together, and that they come and go because we have reversible binding. Now, this is occurring despite there being no explicit force between the different proteins. The clustering requires reversible binding so that the system can evolve, and it also requires bi- or multivalency. So if we include just monovalent factors, no clusters or loops form. We must have at least bivalency so that we can form a bridge and so a loop. And that's why it's called the bridging-induced attraction. So what's driving this clustering? We start with a string of beads that might form a structure like this, and it might have bound some proteins like these. Now let's focus on this particular red factor. It's reversible binding, so it dissociates, but there's a high local concentration here, so it's very likely to be caught nearby. If a pink bead diffuses into this local concentration, it is likely to be caught. So that you can see that this cluster is going to grow as positive feedback drives its growth. And it goes on growing until the number of DNA loops within it becomes so high that it's just too crowded to add any more DNA into the local space. And growth continues until there's about 12 proteins per cluster. So this positive feedback occurs also over here. This green one might bind to the light green beads, and if a red binding bead comes by, of course it won't bind because there's no affinity, but if a green one comes, it is caught. So this cluster will grow. And the two types of clusters are growing in different places in 3D space because we put the pink and the light green beads in different places on the genetic map. So we can extend this approach to a whole human chromosome. And I've shown you 20 beads worth around those genes. So we go to the genome browser and find the active regions and the inactive ones and color the beads accordingly so that red factors bind to the pink beads and black factors bind to the gray beads. And then after we've run the simulations, we find that many of the high C contacts seen experimentally are obtained, as well as TADs and AB compartments. They're all emerging spontaneously without the need for any other input. So this classical mechanism, to summarize, shows that when the transcription machinery binds, clusters, loops, TADs, and higher order structures emerge spontaneously without any energy input. In other words, we've gone from the simple to the complex. In the next section, I'll show that we can also generate small world networks in an inevitable and simple way. Thank you for your interest.